Welcome back again. At this juncture, we'll explore the fascinating world of invertebrates, specifically the animal group from phylum Porifera to phylum Arthropoda. These groups include sponges, jellyfish, flatworms, mollusks, earthworms, and even insects and crustaceans. By the end of this lesson, you'll see how these animals evolve in structure and complexity, from simple body form to highly organized systems. After this session, you should be able to describe the main characteristics of 7 major animal phyla: Porifera, Cnidaria, Ctenophora, Platyhelminthes, Mollusca, Annelida, and Arthropoda. You'll also be able to compare and differentiate their unique features and body plans, so take note of patterns, symmetry, and body organization as we go along. Multicellular and tissue levels of organization, the smaller Lophotrochozoan phyla. Right here is the evolutionary relationships of Porifera, Cnidaria, and Ctenophora under the basal phyla. This figure shows one interpretation of the relationship among them. Two other or members of the animal kingdom, such as those under the Lophotrochozoa, Ecdysozoa, and Deuterostomia, even the one that is on the outside of the bracket, the Protista, which we have earlier discussed. Evidence for this relationship is based on modern developmental and molecular biology. Members of the phylum Ctenophora are the combed jellies. New information suggests that ancestral members of the phylum Ctenophora may be closest to the roots of this animal phylogeny. Members of the phylum Porifera are probably derived from ancestral choanoflagellate stocks, while members of the phylum Cnidaria arose very early in animal evolution, probably from radially symmetrical ancestors. Animals originated from unicellular ancestors that eventually formed colonies and developed specialized cells. This led to multicellularity and the evolution of tissues and organs. As we study each phylum, notice how each step adds a new level of organization and complexity, from simple pores and cells to full organ systems. This figure shows 2 hypotheses regarding the origin of multicellularity. The first one is the colonial hypothesis, where multicellularity may have risen when cells that are dividing protists produced remained together. Cell invagination could have formed a second cell layer. This hypothesis is supported by the colonial organization of some protozoa. The second one is the sinusoidal hypothesis, where multicellularity could have arisen when plasma membranes formed within the cytoplasm of a large sinusoidal protist. Multinucleate bilateral ciliates and developmental patterns of certain insects support this hypothesis. Now, we go to the phylum Porifera, or the sponges. The Porifera, or sponges, are the simplest multicellular animals. Their name literally means pore-bearers. Sponges doesn't have true tissues or organs. Instead, their bodies are made of specialized cells like choanocytes that help filter water and capture food. They are mostly marine and show either asymmetrical or radial symmetry. The body has pores and channels that allow water to circulate, making them filter feeders. Now, Porifera, from the Latin porus or pora, and fera to bear. Basically, they are sponges, which are primarily marine animals consisting of loosely organized cells. There are approximately 9,000 species of sponges around the world. The main characteristics is either asymmetrical or superficially radial symmetrical. There are 3 cell types present in their body: the pinacocytes, mesenchyme cells, and the choanocytes. The central cavity has a series of branching chambers through which water circulates during filter feeding. There are no tissues or organs, meaning they are at the cellular level. The phylum Porifera would include the class Calcarea, wherein these animals has a spicules composed of calcium carbonate. The spicules are needle-shaped or have 3 or 4 rays. They have ascon, leucon, or cycon body forms. They are all marine. Examples are your calcareous sponges. Species examples is the Grantia and Leucosolinia, which will be given and shown later. The class Hexactinellida are the class of Porifera that has a spicules composed of silica and 6 rate. Spicules often fuse into an intricate lattice, cup, or vase shape. There is a zygon or leucon body form found at 450 to 900 meters depth in tropical wastes of the Indies and Eastern Pacific. Examples are glass sponges. The species would include Euplectella or the Venus flower basket. Then we have the class Demospongiae. They are brilliantly colored sponges with needle-shaped or 4-rayed siliceous spicules or spongin or both. They are mostly leucon body form up to 1 meter in height and diameter. Includes one family of freshwater sponges, the Spongilidae, and the bot sponges. Example species are Eucleona and Spongilla. Then we have the class Homoscleromorpha. They are anatomical simple and encrusting in form, spiculous and simple in shape or absent. Occurs at depth ranging from shallow marine shelves to depths of 1,000 meter. Species examples are your Oscarella and Plakina. Now, I will be showing you examples of the Porifera or sponges. The first one is Leucosolinia under the class Calcarea. Then, we have Euplectella under the class Hexactinellida. Then, we have Spongilidae under the class Demospongiae. Then, we have Plakina under class Homoscleromorpha. Now, here shown is the morphology of a simple sponge. In this example, pinacocyte form the outer body wall and mesenchyme cells and spicules are in the mesohyl. Porocyte that extend through the body wall from ostia. The second one, choanocytes are cells with a flagellum surrounded by collar or a microvilli that traps food particles. Food moves towards the base of the cell where it is incorporated into a food vacuole and passed to the amoeboid mesenchyme cell where digestion would take place. The blue arrow shows water flow patterns. The brown arrow would show the direction of movement of trapped food particles. Next one is the sponge body forms. We have ascon, cycon, and the leucon. An ascon sponge is when choanocytes line the spongy cell in ascon sponges. The cycon sponge is when the body wall of the cycon sponges appeared folded. The choanocytes line radial canals that open into spongy cell. A leucon sponge, the proliferation of canals and chambers results in the loss of spongy cell as a distinct chamber. Multiple oscula are frequently present. The blue arrows here show the direction of the water flow. Next, we have the phylum Cnidaria. Moving up in complexity, we have the Cnidaria, or the Cnidarians, or the Cnidaria coelenterata, which would include jellyfish, corals, sea anemones, and hydras. These animals exhibit radial or biradial symmetry, meaning their body parts radiate from a central axis. They are diploblastic, having two main tissue layers, an epidermis and a gastrodermis that would be the jelly-like layer called the mesoglia in between. A defining feature is the cnidocyte or the stinging cells that is used for defense and capturing prey. Cnidarians also have a nerve net, a simple nervous system, and a gastrovascular cavity for digestion. Examples of this would include Hydra, Obelia, which are small, often found in colonies, and then the Aurelia, the common moon jelly, the Metridium, a sea anemone. Some, like Chironex, the box jellyfish, are known for their potent sting. Many cnidarians, especially corals, form reefs that are essential for marine biodiversity. Their main characteristic is that radial symmetry or modified as biradial symmetry, appearing diploblastic